Welcome back to this next video, and in this video we are going to talk about the affinity chromatography. And this affinity chromatography is another direct method that is used in the study of the proteomics. In the previous videos I told you about the protein microarray, which was also a direct method that was used in the study of proteomic study. So affinity chromatography is another method that you can use in the direct study of the proteomics. Now, if you talk about the biomolecules, they are usually purified using different purification techniques, and these purification techniques that actually separate the biomolecules according to differences in specific properties. Now, in the following table, I have drawn some of the, I have given you some of the techniques that can actually separate the biomolecules based on their specific property. For example, if you talk about the affinity chromatography, the technique that we'll be focusing on in this video, that actually is a biorecognition chromatography. What I mean by that is that they can actually separate the biomolecules based on the ligand specificity. If you talk about the ion exchange chromatography, they actually utilize charge is the criteria or the property for the separation of different biomolecules from each other. If you talk about the gel filtration, which is sometimes called is the size exclusion chromatography. So the gel filtration or the size exclusion chromatography is actually separating the mixture of biomolecules from each other based on size. If you talk about the hydrophobic interaction chromatography, they usually utilize the property of the hydrophobicity of the different biomolecules to separate them from one another. So what is chromatography? Now the chromatography is a laboratory technique that is used for the separation of components of a mixture. If you have got a mixture of the different components, you can actually use the chromatography as a technique to separate these components from each other. Now what you do is that you actually dissolve the mixture in a fluid. So that fluid can be gas, that can be solvent. For example, it can be water. So based on the mobile phase, these solvents or these fluids, they are actually called as the mobile phase, and based on the mobile phase there are different types of the chromatography which we are not going into detail, but what you do is that you will be dissolving your mixture in a fluid, and this fluid is actually carrying the mixture through a system on which is fixed on a material which is called is the stationary phase. Now you have got a system. That system can be present in a column, that can be present in a capillary tube, that particular system can be present on a plate or on a sheet. So what the mobile phase do is that it actually carries the mixture through the system on which fixed material, which is called is the stationary phase, that have been immobilized. So you can broadly say you have got two phases in the chromatography. One that is moving is called is the mobile phase. The one that is static and that is not moving, which is fixed to something, that may be fixed in a column, on a capillary tube, on a plate, or on a sheet, and that particular fixed material is called is the stationary phase. Now the different constituents of the mixture, they have different affinities for the stationary phase. So when the mobile phase that is moving the mixture through the stationary phase, so the different constituent of the mixture, they have got different affinities for the stationary phase. So the different molecule, they may stay longer or shorter on the stationary phase depending on their interaction with its surface side. So if the component of the mixture, if there is a particular component in the mixture, they have got a stronger affinity for the stationary phase, that is going to stay longer on the stationary phase. Contrary, if there is a component in the mixture which have got a low affinity as compared to the first one, so it is going to stay shorter on the stationary phase. So the components of the mixture, they actually travel at different apparent velocities in the mobile fluid, causing them to separate. So if you summarize the whole thing, and if you look at the basic principle of the chromatography, you can see that the separation of the mixture in a chromatography is based on the differential partitioning between the mobile and the stationary phase. Now this is just an example to show you, say for example you have got a mixture, you have got these three components, this red one or this purple one, and then you have got this light green one. So what you do is that when you apply your mixture on here, say for example at this particular point, and your mobile phase start moving in this particular direction. So if you see that these red spots over here, they have got stronger affinity for the stationary phase, so they are moving very slowly on this solid support onto which stationary phase that have been immobilized. So this is a solid support, it can be a glass, it can be a plate, or it can be a column, and a stationary phase that have been immobilized over here. So when the mobile phase, it is moving the components of the mixture in this particular direction, so these red spots, they are moving very slowly, which means that they have got low affinity for the mobile phase and they have got higher affinity for the stationary phase. Therefore they are staying for a longer period of time in the stationary phase. If you talk about this purple one as compared to the red one, they have got a high affinity for the mobile phase as compared to the stationary phase. If you talk about this green one, they have much stronger affinity for the mobile phase as compared to the stationary phase. Therefore they are moving very fastly when they interact with the mobile phase. So this differential affinity for the stationary and the mobile phase is actually responsible for separating the component of the mixture from one another. So the affinity that we'll be focusing on is the affinity chromatography. So what this term affinity means, now this term affinity is the degree to which substance tend to combine with one another. Say for example, if there are two things, so what is the degree of tendency or degree to which these substances tend to combine with one another? Or in simple terms, this affinity actually refers to love. So if there are two things, if they have got love for one another, they are going to stick to one another. So the affinity chromatography is a method of separating a biomolecule from a mixture based on highly specific macromolecular binding interactions between the biomolecule and another substance, and we will have a detailed discussion on this, what this another substance mean. So in the affinity chromatography, it's actually separating a biomolecule from a mixture, or they are separating the component of the mixture based on highly specific macromolecular binding interaction between those particular biomolecule and the another substance which we call ligand. So affinity chromatography is very useful for its higher selectivity and resolution of separation when you compare the affinity chromatography with other chromatographic techniques, and these are the advantages of the affinity chromatography. They have got high selectivity and high resolution power of separation. Now, if you talk about the affinity chromatography, this is the list which the typical biological interactions that are used in the affinity chromatography. For example, if you are interested in the isolation of a particular enzyme from the mixture of the enzyme, the ligand that you are using that is actually a substrate analog of that particular enzyme. So is the enzyme, they are very specific in their interaction. So from the mixture of the enzyme, only that particular enzyme will interact with the ligand that you have provided on the stationary phase. If you talk about the antigen, of course they are in very high interaction with the antibody. If you have got a specific antigen and you expose that to a particular antibody, these two are going to interact with each other, thereby separating the antigen of interest from the mixture of interest with the help of that particular antibody. If you are interested in the those particular proteins which have got a polysaccharide attached to it, which we call is the glycoproteins, so for the isolation of the glycoproteins from the non-glycoproteins, for example, you can actually utilize the lectins as a ligand. If you talk about the complementary base sequence, you can actually utilize the nucleic acids. If you are interested in the isolation of a receptor from a mixture of a receptor, you can utilize a hormone, for example, for that particular receptor to isolate or to purify that receptor from the mixture of the receptors. If you are interested in the isolation of a biotin conjugated molecule, or if you are just interested in the isolation of the biotin from a mixture, you can utilize the avidine as a ligand. If you are interested in the isolation of the calmodulin binding protein, so the ligand that you can utilize is calmodulin protein itself. If you are interested in the GST fusion protein, so you can actually utilize the glutathione as an ligand for the isolation of that GST fusion protein. For immunoglobulin, you can utilize the protein A and G as the ligand, and the polyhistidine fusion proteins, they can actually be purified from the mixture utilizing different metal ions that we would see in a while. So these are actually the typical biological interactions that are utilized in affinity chromatography to purify the target molecule from the mixture utilizing these specific ligands. Now, affinity chromatography, it takes advantage of specific binding interaction between the enolate, which is normally dissolved in the mobile phase, and a binding partner or ligand which is immobilized in the stationary phase. As I've told you, if your enolate of interest is an antigen, the ligand that you would be using, that will be an antibody. If your enolate of interest is a glycoprotein, your ligand, that will be a lectin protein. So the affinity chromatography, they actually exploit these specific binding interaction to isolate or purify that particular biomolecule from the mixture. Now, in the typical affinity chromatography experiment, these ligands, they are attached to a solid insoluble matrix. We will have a detailed discussion on the matrix in the slides to come. So in a typical affinity chromatography experiment, these ligands, they are attached to a solid insoluble matrix, and these matrix, they usually are polymers such as the agarose or polyacrylamide, and these matrix, they are usually chemically modified to introduce a reactive functional group with which the ligand can react, forming stable covalent bonds. So you can actually immobilize the ligand on the stationary phase, and then you can use this ligand for the separation of your desired protein or your desired biomolecule. Now, if you talk about the components of the affinity chromatography, the first important component, that is the stationary phase. And this stationary phase is actually made up of three things when you talk about the affinity chromatography. The first component, the first subcomponent of the stationary phase, that is matrix, which is actually a solid support. Now, the matrix that we are using in the affinity chromatography, it is an inert support to which a ligand can be directly or indirectly coupled. And if you talk about the useful matrix materials that are used these days, the most common matrix material, they are the agarose, the polyacrylamide, and the silica gel. But if you are choosing a particular matrix, you should have to choose them on certain characters. So in order for a matrix to be effective, it must have certain characters. Now, the first important character an effective matrix should have, that this should be chemically and physically inert. Now, you should not confuse this for the attachment of the ligand, but when we say that the matrix should be chemically and physically inert, we talk about our biomolecule, that it is not chemically or physically interacting with the that is not chemically interacting, or that is not you can say physically interacting with our biomolecule, so that the identity of the biomolecule, that should not be changed. So the first important property of an effective matrix is that it should be chemically and physically inert. It must be insoluble in solvents and buffers like the mobile phase or the elution buffers, the solvents or the buffer that you are using in the process. So the matrix should be insoluble, because if the matrix is not insoluble in the solvent and buffers that you are employing in the process, that means that your ligand, that will be flowing with the mobile phase, and that will not be available for interaction with the desired component in the mixture. So the second effective property of the matrix is that it should be insoluble in solvents and buffers that you are employing in the process of affinity chromatography. Now the matrix, it should be chemically and mechanically stable, so that the flow of the material, the flow of the mobile phase or the elution buffer should not make it unstable. Now the matrix, it should have the ability that it could be easily coupled to a ligand or a spacer arm onto which the ligand can be attached. So the first important thing is that the matrix should have the ability to interact with the ligand so that it can immobilize it. Now what is the spacer arm? We will come to that in a while. So the matrix should have the ability to easily be coupled to a ligand or a spacer arm onto which you can actually attach a ligand if you are utilizing a spacer arm. So you can directly attach your ligand to the matrix, or you can actually utilize a helper in the form of the spacer arm onto which the ligands can be attached. So in this particular case, if you are using the spacer arm, you are indirectly attaching your ligand to the matrix. Now this matrix, it should exhibit good flow properties and have a relatively large surface area for attachment. What I mean by good flow properties is that it should not impede the flow of the mobile phase, because if it is impeding the flow of the mobile phase, that means that the components of the mixture will not be having a good chance for interaction with the stationary and the mobile phase, and that can actually compromise the process of separation of the mixture of the components. So these are some of the properties of the matrix, of an effective matrix. The second component of the stationary phase, that is the spacer arm. Now this spacer arm is used to improve the binding between the ligand and the target molecule by overcoming any effect of steric hindrance. Now if you look at this particular diagram, this blue one is your matrix, this one is your ligand, and this yellow one is your target molecule. Now if you see over here that the ligand that is in very close interaction with this matrix, and it is therefore not allowing a proper binding site for this target molecule. Now what the spacer arm do is, as you can see, this is the spacer arm. On one side it is attached to the matrix, and on the other side it is actually attached to the ligand. So this particular point, as I've told you, that it should be easily coupled to a ligand or a spacer arm, that should be clarified by now, that this is your matrix, this is your ligand, and this is your spacer arm. Now what the spacer arm do is that it actually provide a very good binding site for your target in the mixture or for your desired biomolecule, because it actually exposes the ligand for the proper interaction with your target. So if you see over here, here you have got three things: your matrix, the spacer arm. So the spacer arm is actually improving the binding of the ligand to this particular target molecule. So this spacer arm, if you talk about some of the examples that are used currently, one of the example of the spacer arm is the cyanogen bromide, the other one is the epoxide, the other one is epoxide with epoxide with C6 acid, or you can use actually the diamine. So the spacer arm, it is actually improving the binding of the ligand and the target molecule by overcoming any effect of the stranding hairs over here. Now the third component of stationary phase, that is the ligand. Now if you technically see, this ligand is the actual stationary phase, because it is responsible for the separation of your desired component from the mixture. But you cannot ignore the importance of the spacer arm and the matrix, because the matrix and the spacer arm, they are actually providing a proper and effective environment for the ligand to interact with its target molecule. So this ligand, it actually refers to the molecule that binds reversibly to a specific target molecule. Now this is very important. The ligand you are choosing that should bind reversibly to a specific target molecule, because if the ligand binds irreversibly to your target molecule, that means you cannot purify your target molecule in pure form. That will be in you can say mixture form with the ligand. So the ligand, they refers to the molecule that binds reversibly to a specific target molecule. Now the ligand can be selected only after the nature of the macromolecule to be isolated is known. That means that when you are going for the selection of the ligand, you should know what kind of the macromolecule you are interested in. For example, if you are interested in the isolation of the antigen, your ligand, that should be an antibody. If you are interested in the isolation of a glycoprotein, the ligand, that will be a lectin molecule. So the selection of the ligand is only possible once you have identified the nature of the macromolecule that you are interested in. Now, when a hormone, for example, when a hormone receptor protein is to be purified by affinity chromatography, the hormone itself is an ideal candidate for the ligand. For the antibody isolation, an antigen that may be used as a ligand. If an enzyme is to be purified, a substrate analog, an inhibitor of that particular enzyme, a cofactor for that enzyme, or a factor that may be used as the immobilized ligand for the isolation of the desired enzyme from the mixture of the enzymes. Now, if you talk about the steps in the affinity chromatography, before going into the steps of the affinity chromatography, the second important component after the stationary phase is your mobile phase. The mobile phase is actually solvent when you talk about the affinity chromatography, and this mobile phase is actually chosen based on the nature of the mixture that you are utilizing. So the two important components will be the stationary phase and then your mobile phase. If you talk about the steps in the affinity chromatography, how the process of the affinity chromatography is carried out, so the affinity chromatography process, it can be separated into three main stages. The first stage is known as the equilibration, the other one is the application of sample, and the third step is the elution. So coming towards the first point, so the first step in the affinity chromatography is the equilibration. Now, this particular mechanism, this equilibration, this is the first step in the affinity chromatography, and this first step is actually to prepare a stationary phase by immobilizing one of the two recognized components on an insoluble hydrophobic polymer such as the agarose. So you have got two recognized components, your target and your ligand. So in this particular case, what you have to do is that you have to immobilize your ligand. So say for example, if this is the matrix, say for example this is the saccharose. So what you do is that you actually add your ligand to this saccharose. So now the ligand that has been immobilized. What this means is that you have prepared the first step, or you have prepared the affinity chromatography for the separation of your target from the mixture utilizing this excuse me utilizing this immobilized ligand. So the first step is the equilibration, and it's actually prepare the affinity chromatography for the process by immobilizing the ligand on the matrix. The second step, that is actually the application of the sample. By that I mean that once you have prepared your affinity chromatography for the separation, then you are going to apply your mixture, or you are going to apply your sample to the immobilized ligand. Now, a crude mixture containing the other recognized component is then applied to the stationary phase. Now, only those that are attracted to the stationary phase will bind to the column, and the rest will simply be washed through the column. What I mean by that is that if this is your sample, it contains a lot of things, it is a mixture of the things. So when you apply this mixture, the only component in this particular mixture that has the ability to bind to this ligand, that will bind over here, that will be stuck in the stationary phase, and the impurities, by impurities I mean the other component in this particular sample, that will be eluted with the mobile phase. So when you apply the sample, the component that is specific for this ligand, that is going to stay in the stationary phase in complex with this ligand, and all of the other material that will be simply washed through the column with the help of the mobile phase. Now, this is the whole story. The first thing you do is you go for the equilibration. You actually immobilize your ligand to the matrix. So you have got the immobilized ligand. when you apply your sample, from the sample the desired component that is going to interact with this ligand, and that will get separated. And the third step that we are going to discuss now is the elution step. And what you do is that as you need this component, if you talk about as we are focusing on the proteins, so we need this protein in the purified form. So what you do is then you go for the elution process, and in the process of elution you actually purify your sample or purify your protein in the pure form. It is not in complex with the ligand that you have immobilized on the stationary phase. Now you can utilize actually different kind of the strategies in the process of the elution. Now the most important strategy, that is the use of the competitors. What you can do is that you can find something, or you can find a competitor that has a stronger affinity to your sample than the ligand. Say for example, if this is your ligand and this is your target, so initially the your target that will be in interaction with the ligand. Now what you can actually do is that you can actually apply something that has a stronger affinity to your sample. So these ligand that you are now introducing, they will interact with your target, and the target is going to leave the attached ligand, and you can actually elute them in the free form now in attachment to this particular ligand. So you have actually initially the target that has been immobilized on the stationary phase, and when you are using something stronger that has affinity for your ligand, so that is going to remove your target from these immobilized ligand, and you can actually elute that from the column. Another strategy that you can use is that you can find something that has a stronger affinity for the ligand than your sample. What I mean by that is that if this is your ligand and this is your target protein, now from outside you are adding something which has a much stronger affinity for this immobilized ligand as compared to your target. So these ligands now which have stronger affinity for these particular ligands, they are going to interact with the immobilized ligand, and your target protein or your target that will be eluted with the mobile phase. Now this particular strategy, that is much useful as compared to the first one, because in the first case what you have to do is that you have eluted your target from the column, but now you have to separate it from this particular ligand as well. In this particular strategy, once you are introducing these ligands which are actually interacting with the immobilized ligand, so your target protein or your target that will be eluted in the free form, so you will not be needing any further kind of the processing. So this is the summary of the whole story. What you do is that you go for the equilibration, where you affinity ligands, they are covalently attached to a chromatography support, and they are packed in the column. The second thing you do is that you are going to introduce your sample to this particular column where you have attached the ligands, and in the third step, these target molecules from the mixture, they are going to bind to the ligand based on their affinity, and in the next step, the sample is washed through the column to remove the unbound proteins, because if you are introducing the mobile phase, that is going to remove all of the unbound components, and only the ligand specific components of the mixture, they are going to stay in the column. Then you are going to apply the elution buffer, the elution strategies that we just discussed, and you will be getting the purified protein that will be eluted from the column. Now in the next video we will be talking about some of the specific applications of the affinity chromatography. So if you like the video, please subscribe to my channel, share it with your friends, and like the video.